All right. I'd like to thank all of you for coming here and Christina for organizing this. Obviously, your support is really critical for allowing us to do what we want to do, and which is to try to find a better treatment and ultimately a cure for this disease. So as you know, historically, the focus in terms of research and developing new therapies has been trying to target KIT, because that is what is biologically driving the majority of these cases. And certainly there's been a lot of progress. 10 years of Imatinib, you know, hopefully that will be 20 or 30 years, but that's pretty good. In addition to targeting KIT directly with drugs like Imatinib, Sunitinib, Sorafenib, and others, you heard about other work by Dr. Dixon that we're doing trying to change expression of KIT and other ways to modulate KIT to decrease the function of that and affect change in this disease. But I think it's important that we look at other strategies. And so what I want to talk about is our work, which is really based on Ron's lab, recruiting the immune system and harnessing the effects of your own body's immune system to fight this disease. So just in the way of background, these are three of the drugs that I think you are all well aware of. We have Imatinib, which is used in first-line therapy, Sunitinib, which is used in second-line therapy. I throw up Sorafenib here, which is another drug that's commonly used in second or third-line therapy. And what you can see here is that clinical benefit is pretty high for certainly for first-line Imatinib, with response rates for major shrinkage in 50% of cases or so. And on average, people benefit from that drug for about 2 years or so. And you do get some benefit with the second and third-line therapies as well. All of these drugs were tested in this disease because they target KIT or PDGFR. They also have other targets as well, so they may have effects upon blood vessel production by the tumor. And they all have other biologic effects as well, which may or may not be relevant in terms of the effects in this disease. Ron kind of showed you how to read some of these curves, and this is just what I call a disease control curve, where at baseline, everyone's disease is under control when they start a drug, and then we can see how things change over time. And for all these small molecules, like Imatinib or Sunitinib, this is kind of what the curve looks like. What you see is you have disease control in the majority of patients for a while, but in the vast majority of cases, over time, patients will develop disease progression, the tumor will grow. And so that's the problem we have with the small molecules. We can continue to try to work to find better small molecules or other strategies to bypass the secondary mutations. But this is a common problem we have in GIST as well as other tumors. One way that we can think about targeting cancer in general and GIST in particular is by maybe thinking about the way the immune system reacts with a tumor. And this is something that we don't commonly think about in GIST or sarcomas. The immune system is obviously very, very complicated, and this is just a cartoon. But kind of the basic components here are, this is the tumor cell here. And to mount an immune response to these tumors, pieces of the tumor, what we call tumor antigens, have to be sent to these special cells called dendritic cells, which present that antigen, that piece of tumor, to the immune system, oftentimes in lymph nodes. And then what you get are these T cells, and as you remember from the last talk, you have the good T cells, the T effector cells. Those are these here, which can go and try to kill the tumor cell. You also have the bad T cells, which we call the T regulatory cells, which actually suppress the immune system. And in general, in immunotherapy, you know, when we try to modulate this system, there are a few ways that we can try to do this. One, we can try to enhance the antigen presentation from the tumor to the immune system so that the immune system can mount the response. We can try to change or enhance the production of an effective T cell response. And third, we can try to modulate the tumor microenvironment, which in many cases is immunosuppressive. So even if you can mount effective T cells, the tumor itself can kind of shut that down. There's been a lot of work in immunotherapy for centuries now, and there are now drugs that are showing efficacy. So there are drugs like IL-2, for instance, which is used in kidney cancer and melanoma. There's a drug which I'll talk a little bit more about called Ipilimumab. And if you look at kind of the disease control curves for any of these immunotherapies, this is kind of what they look like. A lot of patients progress in the beginning. You don't get disease control in a high number of patients. What you get is this kind of flattening of the curve. We call this the tail of the curve, where if patients do benefit, they benefit for a long, long time. And in some cases, these patients are really cured of metastatic disease, and that's been true in multiple studies looking at multiple agents. And just as an example, last year there were 2 drugs approved for melanoma, in large part based on work done in this institution. One drug is something called Vemurafenib, which is one of these small molecules. And this is the survival curve. And you can kind of look at the shape. That's the shape. That's a small molecule shape. A lot of people benefit in the beginning, but ultimately they're going to have disease progression. The second drug that was approved is Ipilimumab, which is an immunotherapy. And that's kind of this dark, actually these two dark lines there. But you can see the curve there as well. This is the immunotherapy type curve. And so ideally what we'd like to do is maybe combine the small molecule curve with the immunotherapy curve and kind of get something like this, you know. And so the question is, can we actually do that by combining small molecule therapy with immunotherapy? This is work from Ron's lab, which he's already gone over, but I think what he just told you was that we know that Imatinib-sensitive human GIST tumors are associated with more of the good T cells and less of the bad T cells, and he just showed that. I don't have to go through this again. And also, if you look at the mouse model, if you look at the tumors treated with Imatinib, what you do see are more of these good T cells within the tumor itself, and that's all these kind of dark spots there, which you don't see in the placebo or vehicle-treated mice. More and more people are looking at the immunologic effects of these various small molecules, and I'm not going to go through that data, but kind of just to summarize it, what they've shown is a number of these small molecules can affect the function of these various T cells, of these cells called NK cells, as well as the dendritic cells, which are important in kind of showing the antigen to the immune system. Some of these small molecule drugs can actually suppress the bad T cells, or T regulatory cells. They can also change the production and secretion of these various cytokines, which also modulate the immune system. They can suppress these other negative co-stimulatory molecules, which this gets very confusing. And then as Ron told you and showed you, they also can lead to reduction of IDO within the tumor microenvironment. And just again, IDO is an immunosuppressive enzyme. It's expressed in some tumors, like in melanoma. And then again, based on Ron's work, in just effective KIT inhibition with Imatinib, suppresses the tumor expression of IDO. It activates intratumoral CD8 T cells, the good T cells, and induces death of the bad T cells. And so, you know, basically what we might be able to do here with Imatinib is actually overcome the immunosuppression in the tumor bed. That's the immunologic effect of Imatinib. It decreases IDO expression, which is important in dampening down the immune system in the tumor. So the question is, how can we enhance the immunologic effects of KIT inhibition to enhance clinical benefit? And the one drug that was approved last year for melanoma is this drug called Ipilimumab. And this is a very complicated drug that modulates the immune system. This is a very complicated slide, but I'll just kind of walk you through it because this kind of helps you understand how this drug works. What we have here is something called an antigen-presenting cell. This is like that dendritic cell that I showed you. This is what shows the antigen to the immune system. And this is that T cell. This is the immune cell. For T cell activation, you need 2 signals. The first occurs when an antigen, which is this orange thing here, is presented by the antigen-presenting cell in the context of this MHC molecule to the T cell receptor. And that's the first signal. When that happens, you get partial T cell activation, and to use an automobile analogy, this is like putting the keys in the ignition, okay? To get full T cell activation, though, you need a second signal, and that occurs when CD28 on the T cell binds to this thing called B7 on the antigen-presenting cell. When that happens, you get full T cell activation. That's like putting your foot on the gas pedal, okay? Now, to make sure you don't get autoimmune disease, to kind of dampen that down, there's something called CTLA4. When you get T cell activation, CTLA4 kind of migrates from this far side of the T cell to what we call the immunologic synapse here. And when it gets here, it actually binds to B7 with greater affinity than does CD28, okay? And when CTLA4 binds to B7, instead of getting T cell activation, you get T cell inhibition. It's like putting the foot on the brakes, okay? So CTLA4 is like the brakes on the immune system. What this drug Ipilimumab is, it's an antibody that binds to CTLA4, and it blocks the ability of CTLA4 to bind to B7. So it takes the brakes away from the immune system, leads to T-cell potentiation, and lets the immune system do what it needs to do to fight the tumor. So kind of on this schema, what CTLA4 inhibition does is it kind of helps to promote the production of this protective T-cell response. What does this do in melanoma? Well, this was the first randomized trial. This was the trial that actually got the drug approved, where patients were randomized to 3 arms. It's a little bit complicated, but this is a survival curve, and patients were randomized to either Ipilimumab alone, which is that green line, the combination of Ipilimumab plus a vaccine against GP100, which is the yellow line, and they were also randomized to the vaccine alone, which is the red line. And basically what you see here is if you got Ipilimumab, with or without the vaccine, you did better than if you got the vaccine alone. And the vaccine in our minds is equivalent to placebo. This is the tail of the curve phenomenon again, right? So you see patients out at 2 years where if you get the vaccine alone, the 2-year survival is 14%. It's increased up to 24% if you get the drug. And so that's the tail of the curve phenomenon. There was another trial where they combined Ipilimumab with placebo or chemotherapy, and you see the same thing. The yellow line is the patients who got the Ipilimumab. They did better than if they got placebo. And here, if you look at the 1-year, 2-year, and 3-year survivals, it's definitely higher if you got the Ipilimumab. So at 3 years, your chance of being alive is 20%. It's not as good as we want it to be, but that's what it is, versus 12% if you get the chemotherapy. Okay. With immunotherapy, the patterns of response we see are different. This isn't like chemotherapy or like Gleevec. With Imatinib, if you get treated with that, you get almost immediate PET response. You get tumor shrinkage in 50% of cases. With this immunotherapy, it takes a long time to work. And so what we've noted here are these 2 kind of novel patterns of response, which we don't see with chemotherapy or with small molecule therapy. So although we can see immediate shrinkage or stable disease, we also see initial growth in tumor, which can occur for 2, 3, or 4 months, followed by stabilization. And we can even see the development of new lesions, followed by disease stabilization or shrinkage later. And again, the data is in melanoma. This is a patient with melanoma who at baseline had these huge tumor masses here on the back, and you can see it on the CAT scan here and here. And this patient got 4 doses of drug over 12 weeks, and at week 12, you can see the tumor grew. If this was chemotherapy, if this was Imatinib, we'd say, oh, the drug's not working. 4 weeks later, though, you can see that this kind of started to flatten. You can see there's less tumor here on the CAT scan. This is at week 12. This is at week 16. And that continued to occur. So by week 72, there was virtually no tumor left on the back. CT scans virtually normal. This is perhaps a more dramatic example of the unique patterns of response to this drug. This is a patient with low volume disease in the liver. You can't even really see it on the CT scan here. But he got his 4 doses of Ipilimumab over 12 weeks. And this is what his scan looked like afterwards. And this is pretty scary. There are new lesions. The liver is clearly sick here. He felt well, though. He clinically looked good, and we opted to just follow him. And with no further therapy, this is what his scan looked like 8 weeks after this one. And it continued to shrink. So immunotherapy, again, is kind of a different type of treatment. So what Ron showed you is that if we combine the combined KIT inhibition with a CTLA4 inhibition in the mouse model, we get better effects than if we treat the mice with either alone. And so based on that data, and so what we're doing there is not just hitting KIT within the tumor, we're also affecting the tumor microenvironment with Imatinib by suppressing IDO. We're also increasing our ability to mount an effective T-cell response with CTLA4 inhibition. And so based on that, we're just about to begin the first trial combining CTLA4 inhibition with a KIT inhibitor. And so this is going to be what's called a phase 1 study, which means it's the first time we're combining these 2 drugs in people, where we're going to use a drug called Dasatinib, which is a drug that does inhibit KIT as well as a number of other tyrosine kinases with Ipilimumab. And as it is a phase 1 study, really we're looking for safety, and we do obviously hope to see a signal of activity as well. On this study, we are collecting a number of tumor specimens as well as blood specimens to better assess what are the immunologic effects of KIT inhibition alone and the effects of KIT inhibition combined with CTLA4 inhibition. Initially, we're going to start at low doses of the drugs and increase it. And for that part of the study, we're going to allow patients with GIST who've progressed on frontline therapy, as well as patients with other sarcomas. There's going to be a 12-patient expansion cohort for which we just want to treat patients with GIST. And for that cohort, we're going to treat patients who've progressed or been intolerant to Imatinib and Dasatinib. This is kind of the study outline, or the schema. And these are the doses that we're going to be testing. But basically, Dasatinib is a pill which is taken orally daily on a continual basis, and the Imatinib is given IV once every 3 weeks. We do that 4 times, and then they get a maintenance dose at 12-week intervals. On this study, we're going to be getting blood work for some of these correlative studies, which are going to go to Ron's lab, the Wolchak lab here. We're also going to be getting tumor biopsies before and after a week of Dasatinib, and then after combination therapy. And those are really important to help us figure out if we're doing what we think we're doing. This study is going to the IRB in 2 weeks. It's a study we're doing in collaboration with the NCI through something called CTEP. So even though I hope to get IRB approval soon, we probably won't be able to start accrual till the summer. That's all I have. I want to thank the clinical research team. These are the medical oncologists who treat sarcoma in our group, certainly Christina and Ron. And again, the correlative studies for this trial are going to be done in Ron's lab, the Schwartz laboratory, and the Wolchak laboratory. So I'll leave it there, and I'll take any questions.